So, good morning, all. My name is Madhura, and I'm a PhD candidate in the Institute of Bioengineering of Catalonia under the Barcelona Institute of Science and Technology here in Barcelona, Spain. And I'm extremely delighted today to present this talk on peptide-based strategies for selective prostate cancer targeting. According to the Globocan survey for the year 2020 by the World Health Organization, prostate cancer marked the highest incidence rate in males worldwide. A key bottleneck of the current treatments is a lack of selective targeting, suggesting that the current systemic treatments might not be effective for all kinds of tumors, which further gives rise to the alarming statement that one drug does not fit all, especially in the case of cancer, which gives rise to the need for targeted therapies and follow-up regimens for each patient as an individualized approach, which forms the basis of personalized and precision medicine. Nanocarriers have been exploited for cancer therapies for a very long time now, in that various different types of nanocarriers have been developed and thoroughly studied for active cancer targeting. However, even after a decade-long attempts at achieving active targeting, we still see that the efficiency achieved is of 0.7%, which is quite low. And upon reaching the actual tumor cells, it even lowers down. With this inefficiency could be attributed to a multitude of biological factors, thereby calling for a thorough study of nanoparticle design parameters in order to come up with an optimal design for selective targeting of the cancer cells, with a view to achieve increased therapeutic efficacy. Within this framework, we devised a selective prostate cancer targeting model by employing two cell targeting peptides known as the WQP, which has a moderate affinity to PSMA, which is a known abundant prostate cancer marker, and the GE11 peptide, which has a very high affinity to the EGFR, which is a known metastasis prostate cancer marker, with a view to achieve super selective targeting of prostate cancer cells by the virtue of the cooperation between the affinity from the GE11 peptide and the avidity from the weak ligand, which is the WQP peptide, acting in cooperation to selectively target tumor cells, which would then result in formulation of dual nanoparticles with specific stoichiometric ratios on the nanoparticle surface, with the rationale of achieving super selective targeting. The objectives that I would discuss for my talk today would specifically involve the studies about effective GE11 affinity, wherein I would discuss different conjugation strategies employed for achieving a desired surface density of the peptide for efficient targeting, and then exploring the WQP avidity, wherein I would talk about the multivalency effect of this peptide nanoparticles over its monomeric form on cellular targeting. So moving on with the effective GE11 affinity, the principal idea of explored here is to see the effect of two different types of conjugation strategies employed, the pre and the post conjugation, which result in different densities of the peptide on the surface, and that would lead to different effect on cellular uptakes of the nanoparticles achieved by each of the strategies. So to describe a bit in detail, the strategy, as the name suggests, the pre-conjugation involves conjugation of the peptide to the polymer prior to formulation of the nanoparticles. So is essentially to obtain a conjugation of the polymer to the peptide following by formulation of the nanoparticles, wherein we employ the process of bulk nanoprecipitation. While the post-conjugation strategy, as the name suggests, involves conjugation of the peptide to already formulated polymeric nanoparticles. So the PLGA pegmalamide in this case, to which the ligand, which is the GE11 peptide, is conjugated after formulation. So in principle, the post-conjugation strategy involves a lot of steps which result in lowered density of the peptide on the nanoparticle surface, and essentially this strategy allows for a lowered amount of control of the surface density. In contrast, the pre-conjugation allows for a higher density of the ligand on the surface in a more controlled manner. So as expected, we see that for the formulated, the pre-formulated nanoparticles, we see almost a twofold increase in the surface surface density of the GE11 peptide. So further, we check the effect of these two formulated nanoparticles from the two different strategies on the cellular uptake in different prostate cancer and healthy prostate cell lines. And we see that in the cell lines with increasing EGFR expression, we see a higher cellular uptake, and also the cellular uptake is found to be increasing from post GE11 nanoparticles to pre-GE11 nanoparticles, which is why we conclude that the pre-conjugation strategy allows for a higher density of the ligand on the surface, and also as a consequence, a higher cellular uptake, which is why it would be the preferred strategy for further studies. Next, we move on to exploring the avidity of the WQP peptide. As I mentioned before, WQP is a weak binding ligand for the PSMA receptor. And here we want to explore, sorry, we want to explore the effect of multivalency of this ligand, in the sense to increase its affinity by allowing for simultaneous attachment of more than one ligands to the receptors. So the idea would be to check the effect of the uptake of the cells of the peptide monomer alone, and along with the peptide functionalized nanoparticles. Again, in this case, we use polymeric PLGA PEG nanoparticles. So to briefly explain the idea of synthesis of the two strategies, we synthesize the WQP peptide in the lab using solid phase peptide synthesis approach, to which we add the sulfoxyphi dye in order to image the monomer within the cells. And so the peptide monomer comprises of the peptide and along with the dye. While for formulation of the particles, we employ again the pre-conjugation strategy. So we conjugate the peptide to the polymer, the PLGA pegmalamide, and then formulate nanoparticles using the process of nanoprecipitation to obtain WQP functionalized nanoparticles. Then we check the effect of these two on the cellular uptake in two cell lines. So we see that with increasing PSMA expression, there is a higher uptake as expected, and in comparison to the monomer alone, we see a higher uptake, almost twofold higher uptake in case of the nanoparticles. But what is the interesting part here is that these nanoparticles also selectively target the cancer cells, which is the 22RV1 here in this case, while sparing the healthy cells, which is the objective of personalized medicine, so as to avoid the systemic effects and have more targeted therapies. So we can conclude that there is a multivalency effect of using a peptide functionalized nanoparticles over using the monomeric form of the peptide, and this results in an enhanced affinity as well as selectivity due to a strong avidity from the weak binding ligands. As a future outlook, the idea is to formulate nanoparticles, dual peptide nanoparticles, comprising of the two peptides that I explained, with a view to selectively target cells with a specific expression levels of the two receptors, and to devise essentially a library of nanoparticles which can selectively target each type of cells individualized, with a view to have a more personalized approach of targeting. So to summarize my talk, I think I have managed to convince you all that the properties of the surface ligands have a crucial role in defining the targeting potential of a nanosystem. The pre-conjugation strategy seems to work better in achieving a more controlled and a higher peptide density on the nanoparticle surface. And avidity, or the combined strength of individual ligands, or the multivalency, seems to increase the overall affinity of a weak binding ligand and improves its selectivity, which is a crucial factor for achieving super selective tumor targeting. With this, I would like to thank my group, the Nanoscopy for Nanomedicine group at IBEC, and also the Severo Ochoa's Excelencia for being my funding agency for carrying out this research, and for all of you for your kind attention. Thank you so much.